to Commissioner of College at Education, Vijayawada. I am Dr. K. Daniel, HOD of Zoology JKC College, Guntur. In this video lesson, I would like to explain about the DNA modifying enzymes, part 1, that is the overview, the types and applications of DNA modifying enzymes. Let me introduce the contents of this lesson. The first one is about the introduction of DNA modifying enzymes. And the second one is the types of DNA modifying enzymes. The third one is the applications of DNA modifying enzymes. And the fourth and fifth headings come under self-assessment questions and web links respectively. So let us see one by one. First of all, about the introduction of DNA modifying enzymes. What are the DNA modifying enzymes? The enzymes which modify DNA molecules are called DNA modifying enzymes. These enzymes are involved in degradation, synthesis, that is the replication, and the alteration of the nucleic acids. So there are three important functions of these DNA molecules. These DNA modifying enzymes. What are they? Number one, synthesis of the or replication of the DNA molecule. The second one is the alteration or change. They can change the nitrogenous base pairing in the DNA molecule. And the third one is the degradation or removing of nitrogenous bases from the DNA molecules. And the enzymes which cleave or cut the DNA sequence at random or specific recognition sites are referred to as restriction enzymes. Restriction enzymes. While the enzymes which are useful in repairing the broken sequence of DNA molecules are called as DNA ligases. So these two enzymes, that is the restriction enzymes and the DNA ligases, both are called as manipulating enzymes. Manipulating enzymes. Of course, these are part and parcel of the DNA modifying enzymes. And the second heading, that is the types of DNA modifying enzymes. So the DNA modifying enzymes can be categorized into four types. Number one, nucleases. Number two, polymerases. Number three, DNA ligases. And the fourth one is the enzymes that cut on DNA termini. So let us see. Let me explain one by one. The first one is the nucleases. Nucleases. What are the nucleases? These are the tools in RDNA technology. They are essential. They are very important and essential tools in RDNA technology or gene cloning. So these are of two types. Number one, endonucleases, and number two, exonucleases. So endonucleases, they are also called as restriction enzymes or restriction endonucleases. What are they, or what are the functions of these endonucleases? These enzymes recognize the base sequence. Of course, these are the restriction sites of the DNA or RNA molecules, and they recognize the base sequence and cleave internal phosphodiester bonds within a DNA molecule. Within a DNA molecule. So these endonucleases, they can cut the DNA molecule within the or internal inside of the DNA molecule. Hence they are called as endo. Endo means internal cleavage or internal cutting of the DNA sequence. There are different types of endonucleases nowadays are available, but like type 1, type 2, type 3, so on and so forth. Out of these types, type 2 restriction enzymes are important tools in gene cloning process. Type 2 restriction enzymes are a very important tool in gene cloning process or rDNA technology. So let me give the examples. You can see here E. coli is one type of E. coli 1, one type of restriction enzyme, type 2, HIND3, BOM H1, DNase 1, etc. Okay, that is about the endonucleases. And the second type of nucleases are exonucleases. The name itself indicates that these enzymes catalyze the hydrolysis of terminal nucleotides either from 5 to 3 prime direction or 3 to 5 prime direction. That is, their action is outside the DNA molecule and exo means outside. So these enzymes, they act upon the DNA molecule, not in the internal inside but outside the DNA molecule. Here you can see the diagram, both endonucleases and exonucleases. You can see here these endonucleases. They are commonly called as molecular scissors because they can cut the DNA molecules. Hence, they are called scissors. What type of scissors? Molecular scissors. So, the endonucleases, they can cut the DNA inside or internal to the DNA molecule, whereas exonucleases, they cut at the terminal region or subterminal region of the DNA molecule. Here you can see the endonucleases, one of the examples, that is HIND3, where you can see these endonucleases or restriction enzymes, they can cut inside the DNA molecule into two pieces. Okay, this type of method is called sticky and cohesive end method. There are two types of methods where the endonuclease can cut the DNA molecules. One is a blunt end and another one is sticky and cohesive ends. So in the blunt end type of incision it is symmetrical, whereas this cohesive end type, these are they are asymmetrical, rather asymmetrical. Okay, so that is about the nucleases. Now come to the polymerases. This is the second type of DNA modifying enzymes. I think you are familiar with this DNA polymerase, another very important tool or enzymes which are useful in the rDNA technology. So these polymerase enzymes add successive nucleotides to growing DNA strands. So almost all the organisms have several types of DNA polymerase enzymes in their body cells. The first DNA polymerase was isolated from a bacteria that is called Escherichia coli. It's a familiar bacteria, you know that? Escherichia coli by Arthur Korenberg in 1976. Hence, this DNA polymerase is also called Korenberg polymerase, which was named after the discoverer. That is the scientist Arthur Korenberg. So every time a cell divides, DNA polymerase role is required because it helps in duplication of cell's DNA so that a copy of original DNA molecule can be inherited to each of the daughter cells. So it is essential, or this DNA polymerase is desperately needed for the duplication of the cell's DNA. So the main function of the DNA polymerase is in the process of elongation of DNA molecule, elongation of double helical structure. These enzymes copy a DNA template strand using base pairing interaction, or RNA by half ladder replication. What does it mean? One half of the DNA ladder serves as a template for recreating the other half during DNA replication. That means one half of the DNA molecule, the double helical structure means it is a ladder-like structure, no? So one of that ladder serves as a template for to create other half during the DNA replication. It is this type of theory was proposed by Watson and Crick. Watson and Crick themselves told this type of process, which is called half ladder replication. So the DNA polymerase can copy the DNA template strand using two methods. That is the first one is the base pairing interaction method. That means nitrogenous base, adenine, thymine, guanine, cytosine, the bonding or the interaction between these nitrogenous base. That is one type, and another one is the half ladder system, as just explained to you. So polymerases almost they have the same function, whether it is a DNA polymerase or RNA polymerase, clinker fragments, reverse transcriptase, so on and so forth. So, what is the main function? They can add successive nucleotides to the growing DNA chain so that the elongation of the chain can be taken place. Here you can see the picturization of the overall direction of the replication of DNA. Here this is the parental DNA. Here it can be forked or it can be duplicated into two separate strands. This is called replication fork, where the primase an enzyme and the replicase and another enzyme, which will help to unwind or unzip this double helical structure. Here you can see two types of strands. Okay, of course this is one parental strand, the blue colored one. The dark blue colored one is the one strand. This strand is between 3 to 5 from or 3 dash to 5 dash end direction. Whereas in the upper region you can see the strand is from 5 prime to 3 prime direction. Okay. Here different types of DNA polymerases are there, like DNA polymerase 1, 2, 3, like that. So I don't want to explain in detail about all these types, because in my next video lesson you can listen or you can learn about these DNA polymerase or different types of DNA polymerase. What they can do, or what the main function already I told you. So whether it is the DNA polymerase 1, 2, 3, or they are useful in the elongation of the DNA or duplication of the DNA molecule. Here what I want to explain, there are two types of strands. In the upper region you can see here, this is called leading strand. The leading strand, the duplication of the parental DNA is taken place from 3 to 5 dash direction, or 3 to 5 prime direction. So from 3 dash end to 5 dash end. This is called leading strand. Whereas in the lower side you can see this is called lagging strand. Lagging strand. In this strand, or this strand extension taken place from 5 dash to 3 dash end. So you remember there are two types of strands while the replication takes place. One is the leading strand and another one is the lagging strand. So the leading strand, it replicate or it extends its template using its template from 3 dash to 5 dash towards the replication fork. Towards the, this is the replication fork, no? Towards the replication fork. Whereas the lagging strand, it can extend its duplication using the parental DNA from 5 prime to 3 prime direction towards the replication fork. So for this elongation, definitely these DNA polymerases are very, very useful along with the primase, primer, and some helicase, other enzyme, and also DNA ligase. It is also one of the DNA modifying agents. All these are necessary for the DNA replication in addition to DNA polymerases. So DNA polymerases are very important tools for the elongation of the DNA molecules. Okay. Next, the third one, DNA modifying enzyme is the DNA ligases. These are the versatile enzymes which facilitate the DNA joining strand together by catalyzing the formation of phosphodiester bond, phosphodiester bonds between two deoxynucleotide residues of the two DNA strands. So, what is the main function of the DNA ligase? Means it can join the DNA fragments. How the DNA fragments are generated? Because of the uh, restriction enzymes. So the restriction enzymes can cut the specific sites or recognition sites of the DNA. Not only it is not necessary, if you want to have gene cloning means it not only needs the DNA cutting but also to join or to add or to join the two fragments which are cut or which are restricted. So DNA ligases play a very important role in joining the two uh, fragments which are cut by the restriction enzymes. Okay. So these ligase or DNA ligase requires energy in the form of ATP or NAD plus for the formation of the phosphodiester bonds. And these enzymes also require a free hydroxy group at the 3' end of the one DNA chain and phosphate group at the 5' end of other DNA strand. Okay, and the role, as I told you, is to seal nicks in the backbone of double-stranded DNA after lagging strand formation to join the Okojokai fragments. What are these? What are the Okojokai fragments? These are the short sequence of DNA nucleotides which are synthesized discontinuously and later linked together by DNA ligases. So there are already short sequences like primers. In addition to that, there are certain short sequence of DNA nucleotides which are to be linked to the main DNA molecule or during the rDNA technology. So this joining process is essential for normal synthesis of DNA and also for repairing the damaged DNA. Okay. So DNA ligases, they play a very important role in joining the cut already restricted fragments. Not only that, they are also essential for repairing the damaged DNA. Next, here you can see the picture regarding the ligation. What is ligation? Ligation is nothing but the joining of two cut fragments or restricted fragments by the DNA ligase. Here you can see the recombinant DNA because of the process called ligation. Now, the fourth one, the enzymes that act on termini. What is termini? It is the terminal part of the DNA. So these three enzymes are very important, which act only on the terminal region of the DNA. What are they? The phosphatases, polynucleotide kinases, and terminal transferase. Let us see one by one quickly. Phosphatases. Phosphatase enzymes catalyze the cleavage of phosphate group. The cleavage of phosphate group from the substrate, that is from DNA, by using a water molecule. By using a water molecule. That means phosphatases, they can remove the phosphate group from the DNA. Substrate means here the DNA. So these enzymes action is totally opposite to the polynucleotide kinase, where they can add the phosphate group to their substrate. Of course, these phosphatases are two types: acid phosphatases and alkaline phosphatases. Acid phosphatases, they can act, their activity is optimal at pH range below 3 to 6, whereas alkaline phosphatase, they can show their activity at about a pH range between 9 to 10. So these out of these two, alkaline phosphatases are most common DNA modifying enzymes, as they are isolated from various sources like microorganisms, tissues of different organs, mainly the connective tissue of invertebrates and vertebrates, and also from human body tissues. The second type of enzyme which act upon the terminal part of the DNA is the polynucleotide kinases. So these PNKs, generally called as PNKs. PNKs. PNKs means polynucleotide kinases. These are the enzymes that catalyze the transfer of phosphate group from the ATP to 5' end of either DNA or RNA substrate. So as I have just told, they can add a phosphate group to the DNA terminal. And they can convert the 3' phosphate or 5' OH ends into 3 phosphate and 5 phosphate ends, which block further ligation by ligase enzyme. So if there is no need of ligation, then these PNKs can convert these two ends into both phosphate group ends. That is the main purpose. And PNKs also use it to label the ends of DNA or RNA with radioactive phosphate group. Radioactive phosphate. The best example for these PNKs are the T4 polynucleotide kinases, which are widely used, which are isolated from the T4 bacteriophage virus infected E. coli. So the T4 bacteriophage infected the E. coli. From this bacteria, these PNKs are isolated. Next, you can see here how this phosphate group, one phosphate group added to the 5' end of that particular DNA molecule. And a third type of enzyme which act upon the terminal part of the DNA are the terminal transferases. These are also called TDT, terminal deoxynucleotidal transferases. These enzymes add single strand sequences to 3' terminus of DNA molecules. The unique property or the important property of this enzyme is to add nucleotides without the aid of template strands. Generally, for the activity of enzymes, there is a need of having template strands. But this enzyme or these enzymes, they won't need template strand. Directly, they can add nucleotides to the DNA terminal. Okay, these enzymes are first purified from a calf thymus gland, a mammal. Calf's thymus gland expressed in lymphocytes. These enzymes, they are also nowadays they are available commercially by producing the expression bovine gene in E. coli. Okay, here you can see the terminal deoxynucleotidal transferase in yellow color round. So they add nucleotide to the 3' end of the that particular nucleotide. Okay, the last part, the applications of DNA modifying enzymes. Already I explained briefly, but let us see one by one some more applications about the DNA modifying enzymes. The first one, the nucleases. As I told, these are the two types. One is exonucleases and another one is endonucleases. So endonucleases are molecular incisors, whereas the exonucleases are used in generating the single-stranded templates. They help to distinguish gene alleles by recognizing single nucleotide polymorphism SNPs. So these nucleases are also useful in DNA fingerprinting technology. Polymerases, as I told, different types of polymerases are there. DNA polymerase 1, 2, 3, 4, like that. And a very important polymerase familiar is the TARC polymerase, Thermophilus aquaticus. It is used in PCR technique, which is to amplify the DNA fragments. And these polymerases also used in formation of complementary DNA by using reverse transcriptase enzyme. And these enzymes also useful in DNA sequencing. And the third one is DNA ligases. As I just told, these enzymes are very important for joining the two fragmented DNA parts, which was cut by the restriction enzymes. So they help to perform blunt and sticky end ligation. And such type of DNA ligase is also useful in DNA designing to treat cancer and also to develop new antibacterial drugs. Phosphatases, they are useful in removing 5' phosphates from plasmid and bacteriophage vector DNA that have been cut with the restriction enzymes. So they are also used in removing 5' phosphates from fragments of DNA before labeling with radioactive phosphates. And of course, the PNKs, they are helpful in eliminating or reducing RNA contamination in the preparation of plasmid DNA. They are also useful in mapping the mutations in DNA and RNA. And they are also useful in RNA sequence. And of course, the last one, the transferases. Terminal transferases play a very important role in evolution studies and adaptation of vertebrate immune system. So that is about DNA modifying enzymes, types, the overview and applications. Okay, learners, and these are the self-assessment questions. You can assess yourself by doing some work upon these questions. So you can find out the answers for these questions. These are the web links. You can search web or net by going through these web links. Okay, thank you. Thank you very much for listening this lesson. So you can learn the second, my second video lesson on the DNA modifying enzymes too, in which I explained about the DNA polymerase. Okay, thank you. Thank you, one and all.